Okay, so thank you for the opportunity to give a talk about our work using Twist technology to enrich 1.4 million single nucleotide polymorphism targets for ancient DNA analysis. This is work with Nadine Roland, who is the director of the wet laboratory at Harvard Medical School, who I worked with on this project. So I'm first going to give you a little bit of background on the application that we're using this enrichment reagent for, which is ancient DNA analysis, and it's the focus of our group right now. So ancient DNA analysis is an approach that tries to get ancient sequences of DNA from ancient human or other animal remains or plant remains. For humans, it starts with a skeletal remain like a bone. This is a petrous bone from the side of the skull. In a clean room where the goal is to get beneath the surface of the bone so that to protect the sequences we're going to obtain from the contaminating sequences of the people who handle them. We obtain a particularly DNA-rich part of the bone, hopefully. We extract about 40 milligrams of powder, which is about a 20th of the weight of a paperclip. We remove and extract the DNA sequences with a DNA extraction protocol that's optimized to remove inhibiting substances as well as the protein and mineral content. And then we make a double or single-stranded library for Illumina sequencing, and we sequence it in one of the high-throughput sequencers that have greatly reduced the cost of sequencing. Because of this series of technical improvements and know-how about what parts of the skeleton to look at and improved methods for extraction and library preparation using very small amounts of starting material and also the great decrease in cost in sequencing, there's been a real revolution in the ability to extract large amounts of DNA from ancient specimens. And the result is this boom in the amount of data that's become available, so that in 2009, there was no genome-wide human DNA data. Since 2014, there has been approximately a 100-fold increase in the amount of data, and it continues to grow rapidly. A key innovation that made this rapid growth supercharged was the deployment of in-solution enrichment beginning in 2015, which was something that our group, Nadine Roland and I, really had a central role in, where we really thought that this would be a way to greatly increase the amount of and number of samples that we could screen. And because large sample sizes are necessary in order to be able to make important inferences about the past, and also because some human samples only have a very small proportion of human sequences, this enrichment has turned out to be a very effective way to both increase sample size and to make accessible samples that would not otherwise be possible or economical to study. So now I'm going to tell you a little bit more about this in-solution enrichment revolution in ancient DNA analysis. The problem is that ancient DNA analysis is really expensive because so little of the sequences that one obtains are human, often less than 1%. Often they're usually microbial from the organisms that colonize the bone or tooth after the individual dies. And the solution we've focused on, and that this talk is going to be about, is enriching the DNA sequences that make it into the library for positions that are informative about human variation, not just human sequences, but specifically also human sequences that are common variants that are very informative about human history and natural selection. So to address this, the background is that in 2013 and 14, in the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany, Matthias Meyer and colleagues were developing techniques for enrichment of ancient libraries. And we worked to adapt a technique they had developed to enrich not the exome, not the Y chromosome, which were the types of targets they were focusing on, not the whole of chromosome 21, but instead to focus on enriching for a set of polymorphisms that are common in human populations that could be informative about history. So we developed an enrichment reagent that targeted 1.24 million single nucleotide polymorphisms. So a complication was that our design, if generically available, could compete with the microarray companies we ordered it from in solution enrichment products, as it allows probes to be replicated and used in many experiments instead of just one. The company agreed to provide arrays with this design under the condition that we use them for ancient DNA analysis only, and not secondarily distribute the reagent beyond the groups that collaborated to produce it. We fully published the probe design with our first paper so others could replicate it, and indeed some groups have. For example, Johannes's group and at the Max Planck Institute for the Science of Human History in Jena, Germany, separately ordered this targeting set. This design that we developed has been responsible for more than 70% of the whole genome human DNA in the ancient DNA literature. So just to show you the impact this has had, this is a wonderful study that came out in 2015, led by S.K. Willerslev's group in Copenhagen in Denmark, which was a study of 101 ancient individuals who lived mostly between 4,000 to 5,000 years ago in Central Europe, Eastern Europe, and the steppe north of the Black and Caspian Seas. So they successfully got DNA from 100 individuals, and they brute force shotgun sequenced them, investing about a billion sequences typically in each sample. And this was an expensive project, but produced a really powerful scientific results. The average coverage after sequencing was about 0.17 average coverage on the targeted positions, but that was enough to make a lot of inferences. In the same year, and around the same time, we published our first large-scale study focusing on the same types of samples, but our coverages were much higher, about 4 times higher on average, and we did about 40 times less sequencing by using this approach. So this shows the power of the method, and both shotgun sequencing and enrichment have advantages and strengths, but we've continued to use for many, but not all of our analyses, this enrichment approach. This is not the only enrichment approach that's been successfully deployed for ancient DNA analysis, and I wanted to highlight a few. The Max Planck group with Matthias Meyer and colleagues have targeted an additional 2.3 million SNPs, focusing on the Y chromosome, also focuses on the Y chromosome, the exome, pathogens. Another 1.7 million SNP positions have been targeted on the from the mega SNP array. Were targeted using the seq-cap-easy technology by Nimblegen, and were published in this PNAS paper in 2018. There's been a reagent that's been developed specifically with the goal of ancient DNA analysis, at least in part, by Arbor Biosciences, and it's been used in 3 published papers in Science and PNAS. We worked with Pontus Skoglund, Ian Matheson, and Alan Cooper to replicate the 1240K target sets plus a module targeting an additional 800,000 transversion SNPs. With Arbor Biosciences, another company, we're actively testing this, and it should be possible for anybody to order this, and I'm excited about that. We don't yet know how effectively it works, but that should be an exciting reagent for the community. And this talk is about the work we've done with Twist very intensively to develop and replicate the 1240K target set and supplement it with an additional 160,000 SNPs. And we are optimizing this, as you'll see, and it should be possible for anyone to order. Affordable large-scale capture reagents are essential to make modern whole genome ancient DNA accessible to many laboratories to democratize ancient DNA, and this is something that our group is really dedicated to, which is why we've been working so hard to make these reagents available. So now I'm going to go over a little bit the bait strategies that we've used, some of them that we've used to target a SNP. So this is a cartoon of a strand of DNA with an adenine cytosine polymorphism. And the old strategy that we've used in some cases was a tiling strategy, where we would have 52 base pair sequences, probes, tiled every 3 base pair over a region of interest, including a SNP. For the 1240K design that we've used so heavily, we used a 4-bait design, which went 52 bases, but not quite including the SNP, 52 bases in the other direction, not quite including the SNP, and 2 probes centered on the SNP, but with the 2 alternative alleles. The advantage of this design was that it didn't bias the capture toward one of the alleles or the other. We wanted to have symmetry for our population history analyses and not cause biases. So we worked very hard to develop a non-biased design. In our new design, which is what Twist is implementing, we have a longer probe, 80 bases, and we actually, instead of putting one of the 2 alleles, we somehow undermine ourselves a little bit by putting a third allele, for example, guanine instead of an adenine and a cytosine at the SNP position. The reason, again, is to avoid bias toward one allele or the other. We randomly choose one of the two alternative alleles. And we know that this slightly reduces our capture efficiency, but we think it's a price worth paying to remove bias. So differences between the 1240K and twist enrichment for ancient DNA. There are 1240K uses 52 base pair probes that equally balance both alternative alleles. Twist uses 80 base pair probes, targeting a third allele to eliminate bias or reduce bias. 1240K uses single-stranded DNA probes in the actual enrichment. Twist uses double-stranded DNA probes. And the 1240K, importantly, relies on 2 rounds of hybridization and capture. In the twist, we only use 1 round of hybridization and capture. That greatly reduces the amount of time required for the enrichment experiment. And we believe it also reduces the amount of bias toward particular positions that comes from 2 rounds of capture. So we had an extraordinary evenness of coverage in our pilot experiment. We took a random set of 50,000, approximately 50,000 SNPs from our 1240K set. And with twist, we made a targeting reagent for those 50,000 SNPs. We then ran them on a bunch of libraries for which we already had 1240K data and shotgun data. And here are the results. So what you see in this plot is a histogram where what you see is the coverage, the number of SNPs or the density of SNPs covered at each coverage as a fraction of the average coverage. And what you see is that the twist experiment produces a really nice tight curve where the great majority of SNPs are within 50% of the average coverage. That's similar for shotgun, and somehow, a bit surprisingly, shotgun is less uniform with more SNPs, very low or very high coverage. We don't understand why that is, but certainly twist seems to be performing well. And the 1240K data from this perspective is really poor in that there are many targeted SNPs that are just hardly being captured. We already knew that in 1240K data, even though 1.24 million SNPs are targeted, we almost never get more than 850,000 SNPs hit, because some SNPs are badly targeted. In one twist library, we hit 98% of the targeted SNPs, compared to 76% for the same exact library analyzed by 1240K. So now I'll tell you a little bit about the content of the 1.4 million SNP twist reagent. It's a clone, basically, of the 1240K reagent plus additional content. So here's the 1240K content. It has all of the SNPs, or nearly all of them, from the Affymetrix human origins SNP array, which has been genotyped on almost 10,000 people from almost 1,000 populations around the world, present-day groups. Almost all the SNP content of the Illumina 610 quad array, a standard genome-wide association study array. Almost all the content of the 55,000 SNP on the Affymetrix 50K array. About 33,000 SNPs on chromosome Y. And about 47,000 SNPs aimed at additional phenotypic content supplementation. What we've added is 56,000 additional SNPs targeting targets on chromosome Y, 95,000 additional phenotypically relevant SNPs from a variety of mechanisms of identifying them, and 40,000 probes to tile additional regions of interest, like key genes where we wanted to be able to detect complex variation, complex polymorphisms, new variants, human accelerated regions, and highly methylated CpG sites for methylation analysis of ancient individuals. The total number of targeted SNPs is 1.4 million, and we understand that this is Twist's largest, or one of their largest designs to date. We performed 9 rounds of optimization with different amounts of baits, hybridization buffer volume, streptavidin volume, input DNA library amount, hybridization temperature, stringent wash temperature. We've tried multiplexing, which is what Twist recommends for its captures, but we really can't see how to effectively multiplex ancient DNA libraries that we don't know the complexities of those libraries coming into it. And if you multiplex libraries of different complexities, the expectation is that that will result in differential amplification of the, it doesn't give enough attention to the low complexity libraries. The current results are promising, but not done yet. We worked on 4 single-stranded libraries with a variety of richnesses and complexities. So here are their percentages of human DNA, ranging from 4% to about 0.2%, so a wide range. We focused on libraries with relatively low percentage of human DNA because that's where capture adds its greatest value. The performance, I would think, should be even better for more complex libraries, but we're testing. And here's the coverages from 1240K capture, which ranged from 1.4-fold average coverage on the targets to 0.005-fold on those targets. So a really huge range of coverage. So one thing that we immediately notice is that when you run 1240K versus Twist, Twist performs much less well from the perspective of the fraction of the sequences hitting the 1.24 million targets. It's only, for example, for the best library, 4% compared to 26%. And that's not because Twist is aiming at a slightly larger target set. That can't explain only a small fraction of it. What's explaining it is the fact that there's only one round of enrichment rather than two in Twist, and we've chosen to do just one round of enrichment. There are benefits to that, though, because the complexity gets reduced in each round of enrichment, and the outcome of this result is that Twist is, in each of these cases, produced higher coverage. This isn't exactly an apples-to-apples experiment. The same number of total sequences were not run. In each case, 1240K, we're pretty sure, saturates at a lower complexity and ultimate coverage than Twist does, but we need to do more sequences per experiment for Twist, both to overcome the low percentage of sequences hitting the targets that we're interested in, and secondly, in order to mine out the additional complexity that we can access with this technology. So the end game for bringing this reagent to maturity, we have preliminary work applying robotic automation on Agile and Bravo workstations. This is tentatively working. We're testing double-stranded libraries, and this is tentatively working. Most of our work has been on single-stranded libraries, but we wanted to also work on double-stranded libraries. We've been spiking in mitochondrial bait sets because we'd like to sequence the mitochondrial genome at the same time. It's clearly working, but we need to get the ratios right. Still largely untested are generating data on many libraries to assess uniformity of target coverage and allelic bias toward one allele or the other, similar to the 50K results that I showed you before. We want to do deep sequencing of the same libraries with both 1240K and Twist to understand coverage as a function of sequencing on multiple libraries to really understand how consistent the different performance characteristics are of the two technologies, although we're cautiously optimistic that Twist is an improvement. We'd like to do population genetic analysis to test if Twist data and 1240K data are associated with biases, with Twist samples seeing process samples seeing more like other Twist process samples and 1240K samples being more similar to other 1240K like samples, process samples. That would be a problem for us because there's so much legacy data produced by 1240K that even if Twist is a more efficient technology, if it can't be easily co-analyzed with 1240K data, that's a severe drawback. So we want to understand if there are biases, and if so, how we can mitigate them. We're optimistic that we'll be switching our own production to the Twist reagent. Twist promised to make the reagent available to anybody interested in using it at a reasonable cost, which we're very happy about. And I want to thank the collaborators from Twist, Mark Honsugar, Ellie Juarez, and Keith McKenna, who made this possible, and people from our group who worked on this, Matthew Ma, Shop Malik, and Nadine Rowland, who really led this work. And many funders provide us with the resources to do all these rounds of testing and so on to make this possible. Thank you.